Well, thank you all for being here today. We have the special pleasure, like every year or half year, to welcome the current cluster-affiliated cohort of the ZUKOnnect Fellows. So welcome, Patrick, Swadhin, and Antonieta, who will present their research today. And without much further ado, I'll let you go. There you go.
Thank you, Alexandra, and hello for everyone. Well, I'm actually doing my postdoc at the UNAM in Mexico right now at the School of Medicine, and this is my postdoctoral project, and actually part of this project is the study that brings me here to the Constance University. So this is the title, C58J, Brain Connectivity and Complexity in a Murine Model of Autism. So I would like to start telling you a little bit about key facts about autism. These key facts are from the World Health Organization, and the first one is that autism, also referred as autism spectrum disorder, constitutes a diverse group of conditions related to the development of brain. About 1 in 100 children has autism, and characteristics may be detected in early childhood, but autism is often not diagnosed until much later, and this rate is growing every year. Also, the abilities and needs of autistic people vary and can evolve over time. And this is why we consider autism right now as a spectrum. Actually, this happened until the 5th edition of the DSM, the Diagnostic and Statistical Manual of Mental Disorders. Before that, the autism was considered more in a linear way, and this is something that is preserved for many people. They think that there are people who are less autistic or more autistic, but this is not actually true. It's a spectrum. So you may have these skills or lack of skills that appears. It's a very complex phenomena. So you can have an autistic person who has very low language skills, but very high motor skills, and maybe is a great dancer. You may have a person with autism with a lot of sensory skills, but problems with the executive functioning. This means they have problems learning, they're having problems with attention and memory, for example. So you will never see a person with autism that is exactly the same that the other one. So all these stereotypes that we have with autism are actually not that true. So this idea of the spectrum actually make much more difficult the diagnosis, but more than the diagnosis, because we have the manual, the most difficult part is the understanding of autism. So all the diagnosis is actually based on behavioral reports, and every biomarker or knowledge about the physiological basis of autism is actually very useful right now to get better diagnosis, early diagnosis, and a better understanding of the phenomena. Also help us for the treatments, right? Because we need to help people to get a better quality of life. Autism is not something that has to be cured. It's a condition. It's a difference of way of living and the way they see and experiment the world. So we need more understanding to integrate them better and for integrate us to them better, right? So this is where it comes, the C58J strain. This is a very special strain of mice because it is an inbred strain. It's not genetically modified. It's not, they don't hurt anything in their brain to get the behavior. And this strain actually has some behaviors very similar to that of autism. We are going to see the behavior of the control. They explore around. They don't have any problem like going here and there. But the C58J shows these repetitive behaviors constantly. They doesn't want to explore, and they also have this stereotyped jumping backwards that this is particular for this strain. So it has awakened a lot of interest in studying this strain. And here we have like the main similarities. The diagnosis criteria of the DMS-5 relies on the deficits in social communication and interaction of people, and also takes in consideration the restrictive and repetitive behavior. That are the criteria for the diagnosis. For the C58J, we can see that these mice have deficit in social interaction, repetitive behaviors, and low preference for social novelty. So that is why it's a very good model to study autism. In Juriquilla, in la UNAM, in Querétaro, there is a 7 Tesla MRI. So we actually did the imaging of these mice. We record the brains of 10 C58BL6 mice that are the controls and 10 C58J mice, all males, because actually there is a lot of discussion about autism in about differences in sexes. So we did this in the Tesla magnetic resonance scanner. You can see it's a very small scanner, but very powerful. So the project objective is, this study aims to construct and analyze functional brain networks of C58J mice using fMRI to uncover connectivity patterns and complexity differences compared to control strain C57BL6. So the first step is pre-processing the data, that this is super tricky and it's taking me a lot of time because actually there is not a public template of the C58J brain. So I have to build it, and this has taken me a lot of time. The second step is actually extracting the bold signals from each region of interest from the brain. So we can, with these signals, we can construct a correlation matrix in time. And with this information, we can actually build the functional network and do some analysis of this data. So this is what we want to do, characterize the functional brain network of this strain and compare these results with those that have been found in humans and see if it is actually a good model, not only behaviorally, but also in brain connections and brain function. So additionally, and this is where this center comes into my story, I want to do some complexity measures, such, in this case, this is my proposal, permutation entropy and range entropy, and also to compute scale invariance in the data of the network. But also I came with Ahmed to see if they have at his lab some more interesting measures or ideas to apply to this network. The potential insights from the research is to get more insights on sensory integration tied to the thalamic olfactory connectivity. This has been seen in these problems of connectivity between sensory and the sensory cortices and the prefrontal cortex has been seen in humans. So we want to see if this happened, especially with the olfactory system in the mice. We want to see how is the network modulation, because there is a theory that actually autistic brains are more local, hyper-connected, and spare connected in large range. So we want to see if this happened to these mice. We want to apply the complexity metrics, because this complexity difference may be serve as indicators for evaluating treatment effectiveness, and the structural alterations in networks may guide preventative ASD approaches. So these findings will enhance understanding of ASD neurobiological basis, informating treatment and diagnostic strategies. This is all the collaborators from here, from Mexico, from different universities. And thank you very much. This is my email if you have any commentary or question.
Thanks a lot. I was just wondering, so they are genetically modified, and you mentioned afterward that there is a lot of inter-individual differences. So how much inter-individual differences is there in this strain? And how this is linked to the, like, so for instance, some of them are making like these jumps and everything. There are some individuals in this genotype that do not jump like that, or less, or
and genetics. What?
Yeah. So, for example, like you say that they have been genetically selected for that, and I was wondering how much then inter-individual variability there is still within this strain.
How much? I'm sorry.
Inter-individual behavioral variability there is in this strain. So if you have some that have much larger symptoms than others that are much less symptoms and things like that, just to try to see the link between the genotype and the behavior in this strain. If it's very, very strict, like it's this genotype, they do this behavior, or if it's this genotype and they have this range of behavior.
Like this genotype, I actually don't know. I actually not sure about that. I have to do more research about it. It's well known for the strain, but I don't have the information right now. It's like, but yeah.
Okay, thanks.
Thank you.
And is it easy to transfer the knowledge about these mice to humans? Because I always learned like that's a big problem.
Yeah.
That many things that work in mice not automatically can be transferred to humans.
The good thing with mice is they actually have the essential parts of the brain that are similar to those in humans. So we can analyze the hypothalamus, we can analyze the ganglia, the basal ganglia, we can analyze the prefrontal cortex, and we can see that more or less the connectivity of this connectivity theory can be applied also from mice to humans. So there are enough similarities to work with it. It's not as complex as human brain, but this is why it has been so much interest in this strain, because we can actually apply some treatments and some ideas to these mice, and it is supposed to work also with humans because of these similarities.
I was wondering, ASD, it's generally considered to be not specifically a mental disorder, but a neurodevelopmental disorder in the classifications.
Yes.
But I think you also mentioned that actually what the anatomical or functional differences between people on the spectrum and not, it's not so well known what actually identifies them at the neurodevelopmental level or the neural level. And then you work with an animal model where you can only look at behavior and the neural level and not so much ask questions and do subjective measures.
Yes.
So how do you know that you're actually on the right track looking at the brain in these bold measures? How do you know that you're actually, with your network analysis, looking at something that's actually happening in people with ASD?
Well, the neuro research in people has been developed in the last years. So we have some features that have been found, especially in amygdala, for example. In the fusiform gyrus, the connectivity in prefrontal cortex, the basal ganglia, and we have very specific zones of the brain that had been spotted in humans that we can actually see these areas in mice. So we will do this comparison just doing that, just like seeing if we can see this in mice that was reported in humans before.
I was just wondering if Antonieta will be focusing on specific regions in her complexity measure.
That's what I'm looking at. This is my very first time working with fMRI, so it's so challenging, but I actually will focus especially on the basal ganglia, because we did like a pilot using TMS. This is another imaging technique. And we saw some differences with basal ganglia, amygdala, and hypothalamus. And we are going to focus on that at the first time to see if we can actually repeat the same results, because TMS, we don't have images on time, so we can see the dynamics of the network. And with fMRI we can. So we can actually, we will actually see if we can repeat those results that we found with the TMS imaging and analyzing.
Thank you so much, Antonieta, for the for the questions. Also, Antonieta will still be around, so if there are any further questions, please feel free to.
Thank you very much for your attention.
You're very welcome.
Hello everyone, I'm Swadhin Agraval. I'm a PhD fellow from India, and I come from this city, Bhopal, which is right at the center of India. And these are my supervisor, Dr. P.B. Sujit, and my co-advisor from here, Dr. India Giovanni Raina. And my host from the ZUKOnnect Fellowship here is Dr. Liang Li. And today I'll be talking about one of our works from the ZUKOnnect proposal about the impact of individual defection in collective motion. So, as we know, like there are a lot of collective motion models that people have tried to model to explain how fishes move or birds flock together. And most of the models, they have really nicely defined set of behavioral rules. For example, in Vishak model, they have this orientation zone where individuals inside this zone of, let's say, particular distance, threshold distance, try to align with each other. And similarly, in a Cousin model, they have these 3 zones of attraction, repulsion, and orientation. However, like, you know, in real world, we know that animals sometimes behave a bit randomly, which people, you know, try to accommodate in their models through incorporating noisy behavior, like some Gaussian noise in the action that they take in terms of rotation, alignment, and different other purposes. But, you know, if you really look into the natural animal groups, it's still like adding noise still means that they're interacting in some way. But we know, like, there are various instances, like, you know, in the fish groups, when, like here, as you see, when they see suddenly some other food sources, or if they sense some kind of attack from any side, they immediately change their direction of motions. And still we manage to see this kind of patterns, as we see here, some kind of coherence in the group. And this is the question that we are trying to ask with this study, that what happens in such systems? How does this randomly, you know, we call this as defecting individuals, those who trigger this kind of sudden motions. So how these randomly defecting individuals influence such collective motion in the groups. So there has been certain related studies in the domain of swarm intelligence and also in the domain of swarm robotics that people have looked into what happens when different individuals are trying to communicate at different frequencies, let's say. So the slower one might look like, you know, less interactive. And some people have looked into, like, what happens if one individual really chooses whom to interact with from the neighbors. And that is another way of looking at it. And some people have also looked into what if there are, you know, decoy or certain agents in the group who try to manipulate the group by acting weirdly randomly. So more kind of an attack from inside the group through some kind of mimicry behaviors. So, but in this study, the way we approach this is we consider a group of individuals in our simulations where each individual can, across the timeline, can defect or cooperate at any given instance randomly. So we fix the probability of when it can switch, like how often it can switch to become a defector or cooperator along the this trajectory that they are taking randomly, let's say, decided through the collective consensus. So, and now here we ask the question that whether they still manage to decide on which direction to move collectively, and can they get really aligned together. So the way we model this is we consider such agents or individuals that can move only in the forward direction with a fixed velocity, and they can only change their directions through interactions. And we use the Dubin's car model for this, which is pretty much common in the simulations. And like the turning rate here, it's constrained by a maximum value, so it cannot rotate exactly the same position. Rather, if it has to change the direction, it has to go forward and take a turn like a car. So, but this turning rate, it is obtained through the interaction model itself. So here, for example, we consider two kinds of models, the topological model and a metric-based model. In the topological model, each individual randomly selects one of its neighbor. It can be at any distances, and copy its direction of heading or the motion direction. And whereas in the metric model, within a certain distance, it looks into like on an average where the individuals are moving, in which direction, and it will try to get to that direction. And here we add an extra layer, which is basically we call this a strategy of cooperation or defection. So if an individual is cooperating, it will obey this interaction rule and try to align. But if it is defecting, it will not obey this interaction rule and keep moving in the same direction. So now what will happen in this kind of a scenario? I start them randomly in this space in 2D. Well, I'm showing it in 3D, but actually we did this study in the 2D. And we ask whether, how much time this kind of a setup takes to get aligned, and how much energy do they spend after alignment. So after they get aligned to, let's say, certain threshold value of 0.8 or beyond, how much energy on an average they spend after they get aligned. So we first did the simulations for the pairwise model, which is a topological model, and we saw that as you increase this probability to defect in the individuals, the time taken to get aligned increases pretty much to a great extent. And the energy is also more or less increasing on an average. But now we switch to the next model, which is the Swadhin model, which is one of the pretty much complex form of the metric-based models. And because we have these 3 zones, there are many possibilities in which the agent can defect. May it defect on the zone of repulsion, so we call this as DZR. May it defect on the orientation zone or the attraction zone, or all the zones at the same time. And there can be other possibilities, but if we kind of understand, like to some extent, how each zones contribute in terms of defection, maybe we'll be able to understand the other combinations. So first thing that we looked into was what is the probability, if I run these simulations for 100 times, let's say, what is the probability that we will see them to get aligned firstly? Maybe like as we see here, when we have a group size of 20, mostly they get polarized or aligned until, let's say, 70% of probability to defect. Especially in the case of zone of orientation, if you defect, they sharply fall when it is 80 or 90% defection. But in case of 50, group size of 50 individuals, we see that this resistance to defection goes down pretty sharply, and after just 30% defection, you cannot even achieve the alignment at all. So this is kind of strange. And another thing to note here is in the type of defection of DZR, you can, you are almost not that much affected by this defection behavior in terms of the group getting aligned. So, but this is like some of the results that I am showing from our simulations. Like in case of 50 agents, we see that if you defect in the zone of repulsion, there is this decrease in the polarization time, and also there is a kind of a minimal amount of energy that they spend at a particular probability to defect. And also when you see in the zone of orientation, it increases. The polarization time increases quickly with the probability to defect, and also the energy expenses of the group increases. So it's really a bad idea to defect in this zone at least. And also in the zone of attraction, we see a similar pattern where, as compared to when you don't have any defectors in the group, the polarization time decreases sharply from, let's say, 30 to 3 time steps. And similarly, there is an optimum in the energy that the individuals spend. And also a similar trend that we see in all kind of, when you defect in all the zones together. And we repeated this experiment with 20 agents, and we kind of see similar trends, but these trends are not that amplified, I would say. They are really obscure in terms of the differences as compared to no defection case. So these are kind of the take-home messages, that in the pairwise interaction model, the energy and time both increases if you have increasing probability to defect of the individuals. And whereas in the cousin model, we see that in these particular zones, if you defect in these particular zones, the energy has a kind of optimal as compared when we think of the probability to defect, and also the time to converge or get aligned kind of decreases. Whereas if you defect in this zone of orientation, the pattern is pretty much similar as the pairwise interaction model. So this is kind of a one-line conclusion that I can say is adharmo api dharmo bhavati yada dharmo adharmo bhavati. So that means like even undesirable actions becomes good when good actions becomes undesirable. And these are like further directions that we are looking into, that why this happens, why the polarization time decreases, why the energy has a certain kind of optimality. So the kind of direction that we are exploring is looking into how the density of defectors in each zones of the individuals is changing. But the question is really not clear whether the density of defectors is something that is affecting it or not. So we are kind of looking forward to it. And further, we have prepared our experimental setup with robots. Currently, we have now 30 of them, where we use the tracking system available here to kind of get a feedback of positions and let them interact within certain zones. And these are some of the preliminary test runs that we did with 4 of them. And we are scaling, we are working towards scaling them up with larger, up to 50 or 60 robots at least. Yeah, and here are certain, the list of our future directions is, these defectors basically are acting like something like stubborn individuals. So others are following them. So we kind of understand it as maybe these defectors can lead to some kind of emergent leadership phenomena in the group if they are placed in certain manner, or if they defect within certain time frame, let's say. So we want to explore how this kind of dynamic role switching can lead to maybe, or maybe not, can lead to emergence of leaderships in swarms, and also explore these strategies, how these strategies are influenced by probabilistic factors and threshold-based decision-making approaches. And finally, we want to test this in the real world with the robots to see how environmental noises and disturbances play a role in such setups. Yeah, that's all. Thank you for your kind attention. And
thank you so much, Swadhin.
Okay, good morning, everyone. My name is Patrick Atagana. I come from Cameroon. I am a lecturer at the University of Ngaoundéré, and I am working on bats. So today, what I'm going to present is some data of my PhD. I was playing with them to see how climate change can alter the future distribution of bat species in the southern Cameroon rainforest. So in Cameroon and most of tropical African zone, forest loss is like the main trait of different ecosystem of animals and the other thing, and this affect the structure of the forest, also the species richness and abundance. What can be the result after that is that we can achieve to the extinction of some species. And for that, I remember I was involved in one project in Guinea with Bat Conservation International, and they want us to make an environmental study to see if one endemic species there in the Mount Nimba will be affected by the mining exploitation. So during our investigation, we saw that the habitat of the species will be modified, and that can be a problem. But what we found also is we discover a new species of bat there. So it means that sometimes we can make some work and disturb, and some species can be lost because we just destroy without know what is there or what is not there. So in this study, this species after 2021, we didn't find it anywhere, even around the Mount Nimba mountain. So why bat? Because bat are an ideal group to study the effect of fragmentation in tropical forests. It's the only group of mammals capable of self-power flight, allowing them to utilize various type of habitat. They have a high diversity after the rodent. In Cameroon, we have 122 species of bat. And also, they have a long lifespan and low reproduction rate. What is also important is that bats are very sensitive to habitat modification. Also, bats are good for our ecosystem because they are contribute to forest regeneration, because with the seed dispersal, also the pollination of some plant. But in Africa, and particularly in Cameroon, the reaction when people are sorry bat is very different. For me, it's easy, I am a bat worker, but for the other population, it's very different. Some of them want to eat the big one. The other are already fear, and what they want to do is just kill them. They don't want to see them. And the actuality also don't help them, because we have coronavirus, Ebola, and other disease. And it's difficult to explain them that bats are not really responsible to that. It's just like supposition. There is no proof that bats are responsible, but they don't really care about that, because like I can say, the image of bat are already so why? What is in your mind? This question was asking by me to my mothers when I told her that I will study bat. And she was like, I spend a lot of money to send you to school, and you come and tell me that you want to study bat. And my response was like, in my mind, I just have bat. So I cannot do something else. And last year, she told me that she really understands, and she's really proud of me, because we discovered a new species in Cameroon also. So that means that there is a lot of work to need to be done in Africa in general and in Cameroon in particular to know which species occur in this country, because we cannot really understand how they are, how we can conserve them without know which species are there and how, which is like their ecologies and with it. So there is a big job. So what I'm trying to do is to trying to conserve bat, try to understand the number of species, which area we can find them. And Cameroon also have this particularity that in the south of the country, we have forest, in the north is savanna, and the far north is the starting of the desert. So we have a complexity of different habitat where bat can be found. So for this, we are assuming the fact that if there is no human activity, the climate change will remain stable. But that is not the case. So climate change is due to human influence. Many studies already prove that, and is influencing and contributing factor to the loss of biodiversity. What we need to do is trying to solve the biodiversity, to save the biodiversity, and also to stop the climate change. My study area is here in the south, is where I collect data for my PhD. And this is the most biggest protected area in Cameroon. And there we can find a primary forest. So wood exploitation and the other mining exploitation is difficult for many companies to arrive there because we have a river around the protected area, and it's difficult for them to go and start destroying. And this is a good place to make this study because in here we have many villages and also many different types of habitats. So there I can find primary forests, secondary forests, cocoa plantation, and also human habitation. How bat can will respond to this different type of habitat? I already publish this result, but what is interesting is that is the different site where I sampled sampling bat during 2 years. So I was going there. It was 14 days each month to capture bat and in the different type of habitat. Yeah, this is the capture site. River border, cave entrance, and here we can see some bat inside the bat. That is the identification setting. So to see how climate change can alter what can be the response of bat to climate change, we use environmental data like in BioClim. Also, we use vegetation data and topography variable to try to predict what can be the response of some of the most abundant species on this protected area. So what we found first, we have this species, Myonicteris torquata. So this, here we have the current distribution. Here, what will be the distribution in 2030, and here what will be the distribution on 2050. So for this species, we know that this species colonize the forest and savanna according to the season. So if we look here, the distribution is more, the distributed area is bigger than in 2030. But in 2050, it will be increased, but it will be like in the right path, going out on the protected area. We have Afronicteris nana. the current distribution. For this species, the distribution area is going to increase with the time. Also, it's it's very present in banana plantation. Maybe we will have many banana plantation in this in this zone. This is Rosetus aegyptiacus, and the distribution seems to stay stable. Diurina cyclops also, you will see here the distribution area will be reduced, but not too much. And this species was the most abundant species of the protected area, but we are seeing that in 2050 the distribution area will be very reduced, and the species seem to be concentrated inside the protected area. And Epomorphs franketi, that is here we have the current distribution, but in 2030 and 2050, I think the species will lose a big area. So what is with this species is also incriminated to be a reservoir for Ebola. So it's also unjust to for virologists. They are killing a lot of them. So and Hyposideros cafe, here we clearly see that the distribution of this species, we are going to lose this species if something is not done. And it's the same for Pipistrellus nanulus. And also for Casinicteris arginis, if something is not done, the species is going to disappear. So, conclusion, we can say that climate change can affect the distribution of bat, and the effect of climate change on bat in this protected area can be positive or negative. But we cannot say that it will be positive because it's not because we have a big number of individuals. It means that it's good for this species. It can be like something to just telling us that because you disturb environment, that's why we have increasing number of this species. It can be like indicator for the disturbance of environment. So in bat, we have some species who are the bioindicator of the disturbance by the increasing number or the decreasing number of individuals. So what next is what I am here for is to study the influence of landscape phenology and climate on collective hosting in short-colored fruit bat. This species occur in the many main city in Central and West Africa. So the treat here is like the people are hunting them for food, but also the main treat is urbanization. This species like to stay around human, and they have big and biggest colony. It's the only migratory species, bat species of Africa, but now the number is going to decrease according to urbanization. So what I am trying to do is like see how landscape phenology, habitat disturbance, deforestation, urbanization can affect the number of individuals of this species in Cameroon, and try to see it in the other main city of Africa, and use those data to convince our government to say, okay, we need to take care and to think on how to preserve this species, because this species is frugivorous, by disseminating some seed. She's also pollinivorous, and she's the one who's pollinating the baobab tree. So if the species disappear, we are going to lose not just the species, but also it will be a problem for forest regeneration, or we will miss some tree species. So, I want to thank the different founders of this project, and also the people who helped me to write the proposal. And here to thank my host, Max Planck, the university, and so thank you for your attention, and I'm happy to take your question.
I have a curiosity about the pollination system. I want to know which kind of plants are pollinated by bats, and if they have any specific characteristics, and if it's necessary to be pollinated at night by bats.
Concerning pollination, we have some tree species who are just pollinated by bat. Like I said, baobab we not found another animal species who can pollinate baobab. We also have I forget the name of the plant in South America. I think it's the one they are using to make tequila. Yes. Yeah.
And it can be pollinated only by bats, or are there any pollinator can contribute?
I think for this species is only by bat, and a specific species of bat is not all the bat species. Yeah.
Thanks for the talk. I have a question about the distribution modeling that you did. So you were comparing the present distribution with 2030 and then 2050. So what was the data for the present that you used? When was it from?
I used the data in my data collection because we travel around the country. And yes, because since 2000, I started working on bat on 2050. So I use those data. And also, what's particular is that the bat we found in forest is not the same we found in savanna. Most of them we have forest species and savanna species. So because we already investigate many protected area and forest zone, we have many data of some distribution species. And we use the coordinate, yes, where we found different individual for each species, put all the coordinate in the model, and the model can say, okay, this place have this characteristic now, and in the future, maybe that will be changed.
Yeah, I'm just curious because, so now we're close to 2030. So do you see this like change in the distribution that you predicted? Do you see it in like the monitoring data that you collect like these days or like in the past years?
Yes, even during this study in 2008, because I started it in 2018 and I finished 2020. But before me, there was another researcher 10 years before me. That's why also I chose the protected area. He did like inventory of bat species of this protected area. So what our data are not the same. There are some species I didn't find, and I found some species with the lowest number of individuals. So abundance was lower than what he found 10 years before. So I also use that to try to see and to see if to verify this we need to keep doing sampling, but it's also costing. We need to have fund for that, and we also need to have people involved to do that, because people are here to work on bats. So it's also a big challenge to.
It's more for the social acceptance. So you mentioned at the end that some people are afraid of bats in Cameroon. And what's the institutional support you would have from the state of Cameroon or whatever, if to protect some species that are particularly at risk or something?
This is why we are trying to work on, because even the government, they are not caring about protect bat. Is also because there is a lack of bat research in Africa, even in Cameroon. When I am the first PhD on bat in the country, and I remember when the COVID started, our minister said that she have like 500 individuals with COVID in the ministry fridge. I was like, it's not serious. So in Africa, most of people know that bats are like bad animals. They have like, they are associated with witch, and everything is bad concerning bat. So they don't have a policy now. So we are trying to work with the government to show them that it's important to conserve those bat species. I remember one time I was trying to send some sample out of the country, and there is like a legalization to pay. You need to pay about the size, the stature of the species, but there was no thing about bat. And they told me that you will pay the same price like for elephant. I was like, it's not possible. Why do you want me to pay? It was like, because we don't have, and it's also a mammal, so you will pay like like it is a mammal, and elephant is a mammal, so you need to pay. I was like, you are not serious. So now they accept, before coming here to work with me, use also UECN statute to try to have our own statute in the country, and maybe that will help for some other researchers when they will come. So there is no law about bat, but we are working on it now.
Thank you, Patrick. That was wonderful, and thank you all today for coming. And I hope you had a good hour. We certainly had, and wish you a great afternoon. Thank you.
Thank you.